person who never made a mistake, never tried anything new. Myself Adiba from Sana Model School. My research is about isolation of enteric bacteria from litopenaeus vannamei. My aim is basically to isolate enteric microbiota from the intestine of litopenaeus vannamei and prove whether it has good or bad bacteria. But wait, what is litopenaeus vannamei? I'll tell you about that. White shrimp is one of the principal crustacean species cultured worldwide. White-leg shrimp, also known as Pacific white shrimp or king prawn, is a species of prawn of eastern Pacific Ocean commonly caught or farmed for food. The main idea behind this is a bacteria capable of producing different extracellular enzymes of potential relevance in the digestive system. The process of isolating microflora from the stomach and intestine of Pacific white shrimp, Litopenaeus vannamei, has proved to be great economic importance in terms of resistance against disease and production of many potential metabolites. This has also proved that the gut of the shrimp has both good bacteria and disease-causing bacteria. The strains of Lactobacillus and Bifidophorans and Bacillus species has good antimicrobial properties. The strains of Vibrio species inside the gut are pathogenic and results in weight loss or death of the species. This results in huge losses in shrimp aquaculture. Thus, isolating the good and bad bacteria will allow us to study about the microbial resistance and extraction of the essential metabolites. The problem which triggered me to do this research was shrimp aquaculture is a huge industry with huge profit. A disease outbreak leads to the huge loss and several environmental degradation. The microbial community of cultured species can play a vital role in aquaculture because it can affect the host growth and survival. My hypothesis on this topic is to check whether there are good or bad bacteria in the intestine of the shrimp, to check whether the good bacteria increases the disease resistance in animals or whether the bad bacteria is responsible for the death of animals, to study the antimicrobial resistance in microflora of shrimp gut. And now, the independent variable of this project is gut of Litopenaeus vannamei. Dependent variable is the growth of microflora, and controlled variable is the nutrient agar, TCBS agar, SS agar, LBMRS agar, timing and temperature. Now, let's get into the procedure. First is the sample collection of healthy shrimp species from the hatcheries. And next is isolation of shrimp gut. Remove the shrimp intestine through sterile forceps. Homogenize the samples. The pH of the samples were taken from the induced media. The pH ranges from 3.5 to 6.5. This shows that the gut can grow even in the highly acidic medium to alkali medium. And now is the sample preparation by the spread plate technique. Preparation of nutrient agar. Take 14 grams of nutrient agar powder and mix it with 500 ml of water. Sterilize the samples in autoclave at 121 degrees Celsius at 80 to 100 atmospheric pressure for 1 hour. Dilution of the samples. Take 1 ml of homogenized mixture of samples and mix it with 9 ml of distilled water. Pour the samples into the plate and rotate it clockwise and anti-clockwise. Incubate the samples. And these are the results of the spread plate method. Now we are going to isolate the pure cultures by quadrant streaking method. And these are the results which I got from the quadrant streaking method. Now let us conduct the antibiotic sensitivity test. Making of the antibiotic disc. Heat the loop and pick few colonies from the agar plate culture and suspend these in the sterile saline. Repeat the same for all the samples. When the suspension is at the correct turbidity, put the sterile disc in saline solution. Now, take some microbial colonies with the help of the cotton swab and gently rub the end of the cotton swab on the agar. This is usually called swab method. Placing of the antibiotic disc. Ensure that you press the disc down onto the agar so that it secures and does not fall. After applying the antibiotic disc, you should aim to get the plates into the incubator within 15 minutes. The zone of inhibition caused with antibiotics is measured after 18 to 28 hours. And these are the results of the antibiotic sensitivity test. And now we are going to conduct the gram staining of these samples. And these are the results of the gram staining of the samples. Now let's come to the discussion. From the spread plate method, we conclude that the growth of microbial colonies was not much visible for the first 2 to 3 days. Later, research showed that growth of the colonies in nutrient agar plates is high on the day 7. And now antibiotic sensitivity test. From the antibiotic sensitivity test by disc diffusion method and gram staining, we obtain the following results. LBMRS agar, I found it to be gram positive. It might be lactobacillus and it's a good bacterium. And the TCBS agar, I found to be Vibrio species. Vibrio genus are one of the most common and widespread disease-causing agent, so it might be gram-negative. And SS agar, I found it to be gram-negative from the gram staining. It may be salmonella. So my hypothesis in this project to check whether there are good or bad bacteria in the intestine of the shrimp has got the results that both good and bad bacteria are present in the gut of litopenaeus vannamei. The good bacteria act as a disease resistant in the shrimp while the bad bacteria causes disease in the shrimp. Hence, my hypothesis has been proved. The applications of this project is when there is a disease breakout in aquaculture by Vibrio species, Salmonella or other organisms which causes disease, lactobacillus can be injected in guts which increase the disease resistance in Litopenaeus vannamei and to avoid death. The outcome of this study will provide a great breakthrough among the aquaculture and emerging industries. Thank you.